So as just said, I did my PhD in Matteo Iannacone's lab, and this is like the result of my PhD there. So I'm not anymore in the lab. I recently joined Yasmin Belkait's lab at Institute-Pasteur. So what I'm gonna show you today is what we published in Nature Immunology a couple of months ago. So basically, what we wanted to understand that, well, Matteo's lab, I mean, maybe some of the ones participating already know, but in Matteo's lab, we are really focused on understanding how the adaptive immune system is able to shape the response against the hepatitis B virus infection in the liver, so the dynamic of immune responses in the liver. So we wanted to understand during my PhD which could have been the role of CD4 T cells in this context, and I'm gonna explain why. So every time that I presented this story, I always wondered which is the best slide to start with, and sometimes I think I always go for this one because I think we somehow don't really remember these in developed countries such as Europe, which is where I live. But HIV infection is still a major global health problem. So this is a global report from last year, but millions of people are still infected with HPV. Despite the fact that we have a prophylactic vaccination in developed countries, there's no cure for chronic HIV infection, which is still a major health problem. So what we know about the HPV basics, just to understand what we've done in this work, is that HPV infects only humans and chimps, and has a super strict tropism only for hepatocytes. So HPV infects only the hepatocytes in the liver, in which it replicates in a non-cytopathic way. And this is the reason why at the beginning of the infection, there's not really a strong innate immune response. So this basically renders the adaptive immune system the main responsible for viral clearance and then healing from the infection. In the last couple of decades, also Matteo's lab contributed in basically showing that CD8s are the main responsible for viral clearance, because they're the ones that once they recognize the infected hepatocytes, they just kill them, and then the infection is resolved. However, these CD8s are dysfunctional in the chronic phase of the disease. So a lot of work has been focused on understanding how CD8 T cells behave in this condition. And in Matteo's lab, they did a really beautiful work in showing one of the possible reasons why, or mechanisms that could explain why we have this dysfunctionality in CD8 T cells during HPV infection. And it's a process that we refer to as intrahepatic priming or hepatocellular priming. So basically, what it was shown 6 years ago in the lab is that basically during HPV infection, CD8 T cells can be primed inside the liver. So this is kind of weird because we know that T cells are usually primed in secondary lymphoid organs, but whenever there's HPV in the liver, CD8 T cells can be primed by hepatocytes, which are not professional antigen-presenting cells, so they're not good in doing this. And basically, long story short, what they trigger is that CD8 T cells acquire this dysfunctional phenotype that is not really overlapping with the classical exhaustion, but still, basically, CD8s accumulate in the liver, but they will not be able to differentiate into effector CD8 T cells and do their job by killing the infected hepatocytes. So a lot of work in the field has been focused, in the HPV field, has been focused on understanding how we can reinvigorate this CD8 T cell response in order to help chronic HPV patients. And one feasible way to do so is finding immunomodulators that can augment the cross-presenting capacities of antigen-presenting cells in the liver in order for them to properly prime CD8 T cells that then are reinvigorated or properly primed from the beginning, and then they're able to differentiate into effector CD8 T cells and then kill infected hepatocytes, leading to viral clearance. So this was the starting point of my project, and we started with a very simple question: what about CD4s in this context? Because CD4s are main orchestrator of the CD8 T cells antiviral response. They usually do that by augmenting the cross-presenting capacity, I mean, modulating antigen-presenting cell activation during viral infections. And we really know almost nothing about CD4s in HBV because they're difficult to study. They're really low in frequency in patients. We didn't have any mouse model in order to study them in the context of the disease. So we really know few about CD4s in the context of this liver infection. So what we did, first thing first, we generated a new mouse model in order to understand which could have been the role of CD4s in this context. And we generated these mice. They are called ENV126 transgenic mice. This was done in the lab by two great technicians, Elisa and Leonardo. Basically, very briefly, in these mice, all CD4 T cells are specific for the surface antigen of HBV. So they recognize a protein, an epitope of the surface antigen of HBV. And we use these mice as donors of CD4s. So, and also they are, basically they're in a RAG1 knockout background. So you only get CD4s that are specific for the virus. So first, okay, we generated the mouse, then we characterized the mouse, everything was okay. And then, again, we wanted to understand whether CD4s could help CD8s. And we thought about, okay, if CD4s can be there and help CD8 T cells, then TH1 will be, I mean, these were TH1, T helper 1 CD4s were the first one that we thought about. So we tried ways in order to get effector CD4s, T helper 1 CD4s ready to help. And what we established in the lab is this pre-activation protocol. This was done in vivo. Basically, we transfer these specific CD4s in RAG1 knockout mice. And one day later, we infect these mice with a VSV that also express the HBV proteins. But well, briefly, if we look in the spleen after 7 days, we get CD4s with a TH1-like phenotype. So you can see that with this pre-activation protocol, we get CD4s that express TBET, CXCR3. They are highly proliferative. They produce TNF interferon gamma upon antigen stimulation and also IL-2. So they had really classical TH1-like phenotype. We confirmed this by also single-cell RNA sequencing. So these CD4s really have a TH1-like signature. So then, okay, we got effectors CD4s. These were ready to help. And we wanted to understand in the context of HBV whether they could have been useful for CD8s. The model that we used, and this is the same that you're going to see in all of the slides, is called HBV transgenic mouse. Basically, in these mice, HBV replicates in the hepatocytes, no evidence of cytopathology, so this is not cytopathic inside mice. Is completely tolerant at the T cell level, so and also hepatocytes are able to produce viral proteins and virions and release them in the circulation. So what I put here is a staining of the core antigen of HBV. This is an histology, just to show you this because it's going to be one of the main readouts that then we use for majority of the experiments. But basically, you can see here in the liver of these transgenic mice, in brown are stained all the hepatocytes where the virus is actively replicating into the cytosol. So basically, in these mice, the amount of hepatocytes that are replicating the virus also will be a readout of let's say, the level of infective level in the liver of these mice. So what we usually do in these mice, we transfer CD8 T cells, CD8 T cells specific for the virus, and these turn out to be dysfunctional. So whenever we transfer CD8s here, they just become dysfunctional and they won't be able to do anything. They will just stay in the liver, accumulate around the big vessels in the liver, but they won't differentiate into effector CD8s. So in this context, we just decided to put CD4s. So what we did, basically, with these mice as the recipients, we transfer CD8 T cells, naive, specific for HBV, and with or without CD4s. Effector CD4s pre-activated, as I just showed you. And well, what we found is that if CD4s are there and are the yellow and blue dots condition, if CD4s are there, we have liver immunopathology that we measure with transaminases, basically is a proxy of liver damage. In the presence of CD4s, we have more effector CD8s. So these CD8s produce interferon gamma, TNF-alpha, granzymes, and also they proliferate more. And you can see here we staying with Ki67, where basically whenever CD4s are there, CD8s are really ameliorated in their function and proliferation. And in general, what we see here is another plot of what we generally see on the surface of CD8s. So there's a general downregulation of some inhibitory markers and an upregulation of activatory molecules. In the tissue, what happens is that if you see the column here, CD8 T cells alone, basically the tissue of the liver is completely clean. So there are some T cells, there are CD8s around the big vessels in the liver. They just stay there, they proliferate a little bit, and they really don't do much more. This is typical of a liver of infected chronic HBV patients. But whenever we add CD4s to the system, then you can see that we start seeing these inflammatory foci in the liver parenchyma. And this is instead typical of HBV acute patients. So whenever we put CD4s in the system, we are able to trigger somehow an immune response in the liver. And on the right, you can see here again a staining of the core antigen in the liver of these mice. And you can see that the amount of brown hepatocytes really decreases in the presence of CD4s along with CD8. So this means that the hepatocytes where the virus was replicating were killed somehow, or at least they died. And same here, we confirm this with Kaspar's staining. You can see that there's some hepatocytes are dying. And the amount of, the level of the viral replication is also reduced, and we check this by either viremia in the serum that goes down, but also by Southern blot. So here you can basically see that all the forms of the HBV genome during its replication are going down whenever we put CD4s in the system. So I, of course, don't have time to show you all the other things that we did to understand which kind of help is this, but trust me, we demonstrated also that this kind of help is heterologous. So basically CD4s can help CD8 T cells of different antigen specificities as long as they are specific for the virus. That this help does not rely on epitope linkage, at least in our model. One of the questions that came just, and also this rely on antigen recognition on MHC class 2. So CD4s need to engage MHC class 2 in order to help CD8. But then one of the main questions that came afterwards was like, okay, where this is happening? Is this something that happens like in a draining lymph node, in liver draining lymph node before, and then we see the effects in the liver, or this is something that is happening locally in the liver? So to answer this question, we performed the same exact experiment, but in mice that were splenectomized and treated with anti-CD62L selecting. So basically in these mice, the naive CD8 T cells cannot recirculate in secondary lymphoid organs anymore, so they can just go into the liver where the virus is. And well, what we were surprised to see is that even though we were preventing the recirculation in secondary lymphoid organs, basically nothing changed really. So there was still liver damage. CD8 T cells were still accumulating in the liver and killing the infected hepatocytes, and there was still inflammation in the liver, infiltration in the liver. So this meant that these CD4s and CD8 were not interacting in a lymph node before coming to the liver and make a mess. And well, then we wanted to understand who was mediating this, because of course CD4s and CD8 need someone else in the middle in order to understand each other. So the main two, we took a look of the main antigen presenting cells in the liver, which are Kupffer cells. There are the resident macrophages of the liver, and dendritic cells. And well, we were surprised to see that in the presence of CD4, so is the co-transfer condition, cases were really like they were much more activated in the presence of CD4s. So we thought, okay, maybe the resident KCs, they are doing the job. So we tried to look for some interaction between CD4s, KCs, and CD8s early on. So one day after we transfer them into the mice, after 24 hours, so a time frame where the priming phase is thought, was happening, is thought to be happen. And we were able to see in the liver, the few events that we were able to see in the liver were actually a Kupffer cell with a CD4 and then a CD8 that are really close by. And interestingly, we were also able to check for these with intravital two-photon microscopy, and same time point, so 24 hours after the transfer, we were able to see these small clusters inside the liver where there were CD8 T cells stuck around a Kupffer cell, and then CD4s like jumping from one KC to another, and seemed like a license in the middle of a licensing process. And in these clusters, we thought that maybe CD8 T cells were really primed in that specific moment because they were really decreasing their speed, and also they were arresting much more nearby these clusters. So basically, they had a phenotype of T cells actively interacting with each other. And then piece of data that was really fun to get. These I'm showing you, I show you mice, but we were able to spot some of these interaction also in chimps. So very briefly, there was this paper that was published more than 15 years ago, and basically what they saw, and actually this was the only evidence that we had that CD4s could have been important during the HBV disease, but basically one chimp was depleted by CD4s before being infected with HBV, and that chimp developed a chronic HBV disease. So this was, at the moment they just hypothesized that it was because CD8 T cells were not properly primed, but however, was speculation. So we were able to get access to those tissues. And here I'm showing you a control chimp, so where everything went well. This is an acute HBV infection in a chimp resembling the one that we have in humans. And at the peak of the infection, during the acute infection, we were able to spot in each of the time point some interaction, like these three cell kind of interactions with a Kupffer cell as a bridge and a CD4 and a CD8. So this might be something that happens as well in humans in a physiological HBV infection. Then last couple of years of this work were like improving, first of all, that cases where resident macrophages were the ones doing the job. And we did this by depleting specifically Kupffer cells in the liver. These are mice that were provided by Martin Guillaume. So basically, KEC4F is expressed only by resident Kupffer cells in the liver. So whenever you give diphtheria toxins to these mice, you take out only cases, leaving all the other macrophages and dendritic cells completely untouched. And this is very specific, so you can see dendritic cells don't care. And in this context, whenever we took out cases from the system, well, the T-cell cooperation was not there anymore. So there was not any more liver infiltration, inflammation, CD8 T cells were not expanding anymore. And also CD4 T cells were not happy anymore because they were not able to expand as usual in the absence of cases. Of course, we checked whether it was the same with dendritic cells, and it was not, because if we take out dendritic cells from the system, nothing changes. So we concluded that cases were the ones mediating this kind of cooperation. And last year, we wanted to understand which was the molecular mechanism. So which were the molecular factors, cytokines involved in this kind of cooperation? Well, good luck with that, I was told, because of course, the way by which CD4s and CD8 T cells cooperate with each other is a mess. A lot of things are involved, cytokines, different receptors, ligands, and signals. So we started from the straightforward choice. So we went for CD4 T and CD4 T ligand because, of course, this is one of the main ways by which CD4 T cells license APCs. And we saw that whenever we blocked CD4 T ligand and CD4 T signaling, everything was abolished. So CD4 T ligand engagement is on top, the first thing that is needed in our model. And then we wanted to understand what was downstream this signaling. And of course, we thought about IL-12. And indeed, what we did is that we took out IL-12 from the system. So we just depleted IL-12 with a blocking antibody, same with CD4 T ligand. And what we got was an intermediate result. So basically, you can see here, this is a control condition. So there's a mess in the liver. Whenever we block CD4 T ligand, everything goes back to zero. So there's nothing anymore. And T cells are just around the big vessels, and there's no liver immunopathology. So no response to HBV. However, when we removed IL-12, we were still able to see some inflammatory foci, but still not as big as the ones that we were able to see in the control condition. The level of liver immunopathology was in the middle, and also same goes for the number of CD8 T cells and their phenotype. So what we were what we saw interesting is that the thing that was resembling the most this kind of intermediate result was the fraction of CD4 T cells. So CD4s, if we blocked CD4 T ligand, were not able to expand at all. And IL-12 gave us some intermediate results. So what we think is that in our model, CD4 T ligand is essential in order to establish this cooperation in the liver. IL-12 somehow produced downstream disengagement, sustains the magnitude of the CD4 T cell response and expansion. And this downstream also influence, of course, the CD8 T cell magnitude of the response. And then here comes the fun fact of the story. Well, in this experiment, I decided to test a new antibody in the lab. I was interested about this molecule. There is SCA1 that we saw in the single cell that these CD4s were expressing. And this was the first time that I saw on the surface of CD8 T cells that SCA1 was really on and off. So before it was like in the presence of CD4s, CD8s are generally down-regulating stuff and then up-regulating activatory molecules. And SCA1 instead, it was really on-off. So SCA1 was there if CD8s were helped and was completely absent if CD8 were not helped anymore. So I thought, okay, whatever is upstream SCA1 must be what KCs are doing that is redirecting the CD8 T cell response. So because of course, we wanted to understand what was downstream the CD4 engagement of the Kupffer cell, because if we wanted, we wanted to translate something in a more translational, let's say, level. So we wanted to understand what was helping CD8s downstream. And well, one of the things that is regulating SCA1 expression is IL-27. So I will be completely honest with you. I really didn't know a lot about IL-27, but the engagement of IL-27 in T cells is able then to trigger the expression of SCA1. And indeed, looking at IL-27 levels in the liver, in this same experiment, we saw that whenever CD4 T ligand was blocked, so when things were not working, IL-27 levels were back to control, let's say, to baseline condition. IL-12 intermediate, but then it was really high when things were working well, like when we had CD4 T cell helping CD8. So we thought, okay, maybe it's IL-27. And indeed, if we block IL-27 and we remove it from the system, everything goes back to zero, like blocking CD4 T ligand, but with a big difference that CD4s don't really care. So basically, whenever we removed IL-27, CD8s were not expanding, CD8s were not differentiating, but CD4s were okay. So the number was the same, the phenotype was the same. So so IL-27 is really something that was downstream the engagement of CD4s, and downstream the communication between the CD4 and the KC. And indeed, if we go back to the experiment where we depleted KCs, basically there's no IL-27 in the system. So these were the experiment where we took out KCs from the system, and whenever KCs are not there, even though CD4s are, there's no more IL-27. So IL-27 was really what KCs were producing downstream engagement of CD4 T with the CD4s. And this was what was really helping CD8s. And to finally test this, we wanted to understand whether we could use and leverage IL-27 alone in order to get some reinvigoration of the CD8 T cell response. And what we did as a last piece of data, so here there are no CD4s anymore, only CD8 T cells. So in this mice, we transfer naive CD8s that are usually, again, they become dysfunctional. But one day later, we give recombinant IL-27 to the system. And well, IL-27 treatment was able to trigger liver immunopathology. CD8s were really expanding, especially the ones that were producing effector cytokines like interferon gamma and enzyme. And you can see that in the liver, there was a huge infiltration of T cells in the liver, and hepatocytes were killed, and the viral replication was inhibited. So IL-27 could be, was able to ameliorate the CD8 T cell phenotype in our model. And final piece of data, we wanted to see whether this could have been useful also for patients. And we collaborate with Antonio Bertoletti's lab at the Duke News in Singapore. Basically, what they did, they isolated PBMCs from chronic HBV patients, and they put these PBMCs in culture in the presence or absence of IL-27 and stimulated with HBV peptides, a cocktail of HBV peptides or CMV peptides as a control of memory response. And then 10 days after, there's really a specific enrichment of T cells in this kind of assay, and they just measure the interferon gamma production on T cells via ELISpot. And what we saw is that was really specific. So IL-27 was able to ameliorate, to stimulate the interferon gamma production by T cells only T cells specific for HBV, and this was not true for CMV T cells, which was great because it means that IL-27 alone cannot trigger alone a memory response, which is a side effect that is, we know that is usually another type of cytokine-based treatment with, for instance, IL-2 or IL-12. This enhancement was true against basically potentially all of the proteins of HBV, so the surface protein, but also the core protein and polymerase, and was specific for CD8s. So CD4s really didn't care, but CD8s were the ones that were really enhanced in their interferon gamma production. So with this, I'm done. This is the final model. So what I show you today is that basically what happens in chronic HBV infection is that CD8 T cells are dysfunctional. One reason why this happens might be that CD8 T cells can be primed inside the liver, and this triggers the establishment of a dysfunctional phenotype and program of the adaptive immune response in the liver. However, if we provide those CD8 T cells as CD4 T cell help, in our model, this CD4 T cell help was mediated by CD4 T engagement on the surface of Kupffer cells in the liver. And this triggered the production by resident Kupffer cells of IL-12, which sustain in a positive feedback loop the response of CD4s, and IL-27, which instead, along with antigen presentation, then is able to revert and ameliorate the HBV-specific CD8 T cell response. And with this, I'm done. This is the lab. So this was a huge effort. A lot of people were involved. So Matteo, I have to thank Matteo because was my PhD mentor, was the one that trusted me with this really amazing project. And Krista and Chiara were our postdoc and an incredible technician in the lab. Without them, I wouldn't be here today presenting you this data. And then a lot of other people that are still in the lab, not in the lab anymore, but also collaborators worldwide were involved.